Great. So we are ready to begin here. Thank you all for joining again today. Welcome to the first session of Bioinformatics for Precision Oncology. Precision medicine is changing the way we understand, diagnose, and treat cancer. The transformation is driven by high throughput molecular data from patients, animal models, and large-scale cell line experiments. In this program, we will explore how high throughput data can be analyzed to study the genomic background of cancer omics data that describes the basic biology associated with cancer onset, development, and outcomes, and see how large-scale datasets collected from clinical trials provide an opportunity to better understand this condition and improve the precision of clinical oncology. My name is Beepsha Biswas, and I am the OmicsLogic Community Manager, responsible for daily interaction with thousands of users who are part of our bioinformatics community managed by Pine Biotech. For those of you joining us for the first time, I wanted to share about the OmicsLogic programs that are a result of a close collaboration between Pine Biotech, an innovative big data analytics company based in the US, and numerous academic institutions that have participated in content evaluation, curriculum review, and project design. As a result, the OmicsLogic programs have been thoroughly tested around the world and are known to lead to excellent outcomes for students with minimal bioinformatics background, including biology and biomedical studies majors, as well as faculty interested in bioinformatics, data science, and biomedical research. OmicsLogic is an international program that is running in 5 different regions with over 10,000 users around the world. Due to this fast growth, our team is working with local and regional coordinators that are helping refine local program logistics and leverage our online training resources, adapting them to the needs of students and researchers around the world. The goal of the OmicsLogic programs is to provide training in bioinformatics, enabling independent research guided by mentor and peer example, developing a growing community with shared passion for data-driven research and appreciation of citizen science, enabling students, clinicians, and faculty of all backgrounds to develop novel and independent research using latest technology in life and data sciences. We will combine online sessions like today, self-guided study materials, and practical assignments for an immersive experience that has proven to be effective in our well-known and respected OmicsLogic programs. The program will contain online sessions, courses on the basics of various types of data like genomics, transcriptomics, etc., as well as project datasets that come from high impact publications on various aspects of cancer biology. So the first platform that I would want you all to get started with is learn.omicslogic. So the platform is learn.omicslogic.com. I'm posting the link to the platform in the chat box. This platform comes with a variety of resources. So let us go ahead and check what this platform is about. So let me go ahead and show you. So this is the Learn platform, and I'll post the link again in the chat box so that you all can go ahead with me and start with this platform. So you'll find that there is a login option here. And once you click on the login option, it will take you to the sign up page. And here you have the option to create an account either with your own email ID. So I'm already logged in, or you can also use any of the social logins. So once you are logged into the platform, you would be directly going to the courses. Now we have added all of the courses here into different lessons. So if any of you were already familiar with the Edu platform, there we had courses divided as Introduction to Genomics, Genomics 1 and 2. What we have done with the Learn platform is added the introductory courses, but also added all the several different stages of the courses into different lessons. So you can start with just lesson 1 and progress. So all participants will have full access to the platform and to all of the courses. So any of the courses marked with a star that you see here are premium courses and can be accessed either as part of the program or via a license, which we are going to be talk about at the very end. So there are a lot of free resources as well for everybody to get started with. So please feel free to explore this platform. And we do not only have courses, we also have projects like these, as well as we have integrated coding in R and Python as part of this training program. So without much ado and waiting for you to get started, what I'm going to be introducing you next to is the server platform. So I hope that you are logging into learn.OmicsLogic. So the server platform is the next resource that all of you will have access to as part of the program. And we are going to be talking more about how to utilize this platform during this entire 1 month training. And for our OmicsLogic participants to reach out to us, I'm sorry about this one, we have a Slack channel. So this channel is only open for all of our participants. And the participants can actually go ahead and join this group. So we will send out personal invites to all participants. This will just make sure that you're able to connect with our team as part of this full 1 month training. So please make sure that you are registered for the program and you're receiving all of these emails from our end. And there are also 2 other groups that I would like to invite all of the participants and everybody who joins any of our programs to. So we have a group on LinkedIn where you can connect with other members from all around the world. They are clinicians, faculty, students, also administrators. So please feel free to join any of these groups. And also a group we have is on Facebook. So you can find us on either of these social media channels and follow us there. Now, let me go back to the presentation quickly and introduce you to our trainers. So we have Dr. Harpreet Kaur, who is a certified OmicsLogic trainer and expert bioinformatician. She will guide the hands-on sessions and assist participants with projects and analysis during the program. Her area of specialization is in cancer genomics and machine learning. Dr. Mazumdar is our OmicsLogic project mentor and an expert in big data computational analysis. He has a PhD in computational biology and over 10 years of experience working with industry and academia. Elia Brodsky is a co-founder and CEO of Pine Biotech. In his current role, he has extensive experience working together with industry and academic partners who are applying multi-omics integration and bioinformatics to a variety of biotechnology and clinical applications. We also have Clinton Kuna, who is joining us as one of the bioinformatics trainer for this program. He has a master in bioinformatics from the University of Guelph and a master's in cellular and molecular medicine from the University of Ottawa. Now I would like to invite Dr. Mazumdar to share more about the OmicsLogic Precision Oncology program. Over to you, Dr. Mazumdar. Thank you, everyone.
Thank you, Deepsha, and hello, everyone. So just a second, let me share my screen. Hello, everyone. Thank you for joining the session, and my name is Mohit Mazumdar. I have done my master's in bioinformatics from Jamia Millia Islamia, and I did my PhD and major research work at Jawaharlal Nehru University, India. And then after finishing my PhD, I was able to work with a bioinformatics company, helping pharmaceutical companies to work who are working on different bioinformatics challenges related to big data and applying machine learning and data science applications of data science in research. So in this current role, I'm working with universities and I'm working with students and research interns to be able to work on several of bioinformatics research problems that we are seeing. And also I'm contributing in data science related curriculums and biological research of at different levels of education. So talking about the session today and about the program and the context of this program. So one of the major objective of this OmicsLogic program is to be able to learn from the asynchronous materials, different and the application of different omics technology, and also learn from the example projects. However, OmicsLogic Oncology research program, the one that we have designed, helps researchers and participants to not just learn from the examples and learn about the different omics technology, but to also explore the hugely available public data on NGS cancer research. But however, I mean, oftentimes it's kind of like really hard. We find it like really hard to find the right kind of omics data for maybe precision oncology research projects. So learning about the impact of NGS and this explosive growth of publicly available data, one just wonder, like you just have to wonder about that, how much data is available and where you can find the RNA-seq data sets on cancer, and start exploring what kind of experiments are have done to answer some of the important questions. That sort of data is which is being utilized by the pharma industry and the healthcare sector. So we kind of have compiled an post here where you can find good data sets and work on and look at the available resources that have generated on, also the scientists who have generated and the research labs who have generated these data sets. So during this time, interacting with many students during our like OmicsLogic educational programs, we realized that the need for high quality data sources that anyone can learn from and use. So defining good data. So good data is a data from collections that follow a certain level of metadata annotation with minimal restrictions and easy access to the files. So for example, details of phenotypic information associated with samples, as well as the file size or sequencing instruments that are being used, those are all important factors. So another criteria would be the number of replicates, where they have whether they are technical or biological. So some of the best repositories that answers these questions and like have those samples are compiled in this blog post. So I'll just let me share this link with all of you so that you can visit this blog post. There you go. So as I was telling you that working on example data sets, so some of the example data sets that are already available, and we will be talking about those projects is like modeling precision medicine. So working on cell line data sets and then working on example data sets, followed by some of the other repositories, like in one of the upcoming workshops that will be happening in April in collaboration with All India Institute of Medical Sciences. We'll be working on TCGA lymphoma data, and then going through the publicly available data specialized for a department of pathology who are working on patient samples related to that. So some of the applications can be directly, you can find directly on publicly available data, which will be utilized in this program. So we'll be really happy to for you to explore these data sets and come up with your own research questions, and we'll be happy to work with you on your research problems throughout this program. So with this information, I'd like to pass it on to Clinton who is here and who will be one of the OmicsLogic trainer to talk, helping us learn about the different aspects of oncology research. So over to you, Clinton.
Thank you, Dr. Mazumdar. Let me
Yeah.
share my screen. All right. So precision medicine is changing the way we understand, diagnose, and treat cancer. This transformation is driven by high throughput molecular data from patients, animal models, and large-scale cell line experiments. Today, we will introduce you to the Bioinformatics for Precision Oncology program. But before we do that, let me share a few points about my background and the reason why bioinformatics is central to the translational research in oncology. My name is Clinton Kuna. I completed my bachelor's degree at Carnegie Mellon University, where I discovered my passion for research. Later on, I decided to continue learning about life sciences. As a result, I now have 2 master's degrees, one from the University of Guelph and the other from the University of Ottawa. Since I was interested in translational bioinformatics, I did a master's in bioinformatics and another one in molecular medicine. As a result, I am familiar with biomedical research as well as data analytics. So cancer is an extremely heterogeneous disease. Even though the war on cancer was announced 70 years ago, brought upon many advances in our understanding of the basic mechanisms of tumor growth and metastasis, it is also clear that the current research did not solve the problem. On the contrary, we are learning more and more about this complex condition. To show how complex cancer is, let me give you an example from my past research experience. There are several proteins that play different roles within certain contexts. For example, ST20-like kinase, or SLK, is a protein that plays an important role in cellular processes such as proliferation, migration, and apoptosis. It affects embryo development and is shown to play a role in tumor genesis. This was studied in a new induced mammary tumor model within mice. These experiments showed that SLK knockout mice developed tumors faster and showed an increased expression of SOX10, an important biomarker in cancer. However, in my own research, we studied SLK in mouse embryonic fibroblasts, where SLK knockout reduces focal adhesion turnover. It turns out that SLK plays different roles depending on the context. Even though this contradicts the other study, it also shows how complex the biology of cancer can be and how careful we have to be with experimental design and data analysis to truly understand its pathogenesis. Research on SLK is ongoing. This is similar to cancer. Every cancer is different, and it's a challenge to determine where those differences exist. We need more advanced methods to determine why a knockout reduces a process in one context, but increases it in another. My master's thesis gave me a glimpse of how bioinformatics can be used to study oncology, and the skills I acquired enabled me to use more sophisticated models and methods of analysis, such as machine learning. Therefore, based on my first master's, I decided to pursue a master's in bioinformatics to learn those complex skills. Pure biology is good, but having the data analysis skills to create meaningful relationships definitely solves major problems that can be translated into applications within medicine. Bioinformatics has exposed me to other fields due to its interdisciplinary nature, such as statistics, engineering, and mathematics. So now let me pass it on to Dr. Harpreet to talk more about her research and her experiences.
Thank you, Clinton. Let me share my screen. Hello everyone, my name is Harpreet Kaur and I have a PhD in Bioinformatics. I have done my doctoral from CSIR Institute of Microbial Technology, India and JNU, New Delhi. Let me give you some glimpse how bioinformatics can be helpful in identifying biomarker or signature for a specific malignancy by taking some examples from my research work. My PhD research was on biomarker discovery for diagnosis and prognosis of the liver cancer using bioinformatics. So for the first part of this project, we used large-scale transcriptomics data to identify a diagnostic biomarker panel for the hepatocellular carcinoma, which is primary liver malignancy. Here we employed 30 datasets containing nearly 4,000 samples to elucidate transcriptomic signature for the diagnosis and the prognosis of the liver cancer, particularly hepatocellular carcinoma, using various bioinformatics techniques. In the second part, we employed 9 datasets containing nearly 350 cholangiocarcinoma, 133 adjacent non-tumorous, and 90 hepatocellular carcinoma samples. So here we scrutinize a 3-gene based signature which is specific to the cholangiocarcinoma, implementing various data mining techniques like random forest, SVM, extra tree, logistic regression, etc. In the next part of our project, we use transcriptomics and methylation data of the hepatocellular carcinoma patient to identify early and the late stage patients. So here we use TCGA data of 350 SCC samples and elucidate 51 features that include 30 RNA transcripts and 20 methylation CpG site to discriminate early and the late stage samples of SCC. So in my current role, I'm OmicsLogic bioinformatics trainer with Bioinformatics. I'm assisting participants to learn about data analysis, machine learning, and visualization in application of large data set used in the biomedical research. Besides, I'm involved in designing different research projects using publicly available data. So to provide hands-on training and to make participants familiar with the approach to design and complete a research project. We are continuously designing different research projects based on publicly available data, which are published in well-known journals. Thus, we have a collection of research projects where you can learn how bioinformatics can be applied in the field of oncology using these freely available datasets to drive biological and clinical important biomarker for a specific cancer conditions. So let me give you a glimpse of one project where we focused on identifying molecular signatures that are associated with the clinical factors of the liver cancer patients based on integration of multi-omics data. Here we explored different omics layers, that is RNA, miRNA, and clinical and demographic characteristics of the SCC patients to find out what are important molecular signatures that are associated with those clinical phenotypes like tumor stage, risk factor, age, gender, etc. So there are a number of the research fellows who have completed their research project in the field of oncology with Pine Biotech Research Fellowship Program. One of our research fellow, Ms. Simay Dolanar, an undergraduate student from Turkey, recently completed her research project entitled Long Non-Coding RNA as Therapeutic Target in CLL, that is chronic lymphocytic leukemia, using publicly available data and implementing various tools that are available on TBio server. Then Ms. Ursha Parekh, who is postgraduate student from India, completed her research project in identification of biomarker to differentiate between neural stem cell and glioblastoma stem cell using various tools on TBio server and employing data set taken from the public repository. Next, Mr. Calamari Marul, who is a PhD student from city in Mexico. He has completed his research project entitled Classification of Triple Negative Breast Cancer Cell Line by Gene Expression Analysis of Transmembrane Protein using Machine Learning Algorithms, based on available cell line data and using different tools from our various platforms. I'm looking forward to being your mentor in the upcoming OmicsLogic pre-season oncology program. Now I would like pass this session to Elia. So over to you, Elia.
Thank you, Dr. Harpreet, and everyone else that had a chance already to present. And I want to pick up on where all of this kind of leads us to, which is the program itself. So up until now, you've seen how to gain access to the different resources, to see where you might see yourself after you complete this program with an independent project or a better understanding of how bioinformatics could be applied to oncology research. And also some examples of real data sets that you can have access to as curated data sets, or then potentially a project of your own where you have to identify a new data set to do your own research. So what I want to speak to you today about is the subject of today's session, which is the introduction to this program. But before I do, let me introduce myself. My name is Elia Brodsky, and I've had the privilege of running hundreds of online and on-site programs related to bioinformatics. And we've done this with our collaborators at universities such as Georgetown University, Louisiana State University, Tulane, University of Haifa, and many others. And so as a result, I've seen thousands of students from all backgrounds complete their training and get empowered by bioinformatics tools to participate in research studies, develop data-driven hypotheses for their projects, and go on to publish or present their projects in poster review sessions. And so what I hope for all of you is that this program will also help you in your journey, whether you're only starting or have already vast experience in oncology. So we hope that this program will be an experience that will provide you with new opportunities and skills that will benefit you for years to come. So in this first session, what we want to do is kind of set the stage for why we're learning about oncology and what are specific characteristics of cell and DNA and omics data that will be used in the future sessions. So we'll talk about challenges associated with big data in cancer research. And then we'll talk about how next generation sequencing data and other types of high throughput data could be used to understand the biological processes that lead to clinical manifestations of cancer. So first, let's talk about what is cancer, because cancer is one of the leading causes of morbidity and mortality worldwide, with almost 10 million people dying from it per year. The cancer prevalence is the greatest in the Western world, as you can see on this map, where you have close to 6% of all people that have cancer during their lifetime in the US. And worldwide, there's an approximately 40% of men and women that will be diagnosed with cancer at some point during their lifetime. And in 2020, an estimated 17,000 children and adolescents ages between 0 and 19 will be diagnosed with cancer, and ultimately over 1,700 young people will die of this disease. And so what we can see from this data is that cancer risk increases dramatically with age. But there are other also causes of cancer that include abnormal weight, use of tobacco, exposure to radiation, as well as many different types of viral infections. In fact, about 5% of all cancers are caused by pathogens, by viruses. So the bottom line is that cancer is a genetic disease, and it is caused by cancer causing or driving genetic changes. Inherited mutations play a role in about 5 to 10% of all cancers, and the rest are associated with somatic mutations and changes in the regulation of cell development. So when we study cancer, we essentially can think about this as a disease of cells. And we can observe the changes of cells from being normal, healthy, functioning, that are similar to other cells in the tissue, to becoming abnormal or deregulated cells that start to form tumors or abnormal growth and metastasize or spread to the other parts of the body. And so we can see how this process takes time. And so there exists an urgent need to identify new biomarkers or signatures in biological processes that can help us with early detection, prognosis, and appropriate treatment selection. And so this is what this program is going to be all about. So in this session, which is called Introduction to Oncology, we'll talk about some of the biological changes in organs, tissues, and cells. What are some of the molecular factors that are associated with tumor development, growth, and metastasis? And how can we view the relationship between clinical diagnosis of this disease and the molecular data that essentially causes this disease to appear and has to be studied in controlled experiments, oftentimes with an association of what we have available for the clinical patient level data. And so this part of the program is essentially a fundamental introduction that many of you are probably already familiar with, but you will kind of have a refresh of some of those terms that we will be using throughout this course, and hopefully that will help us think about the significance of the data and the analysis methods that we will continue to use in the future sessions of this program. So since we talked about cells, one of the primary things that cells go through are individual cycles of cell division. And so cell division is essential for normal growth and normal kind of development of an organism, as well as in our, after we kind of grow up and we mature as adults, cell division is a continuous process. And so it is essential for life, but it must be controlled. And so when a cell divides, it goes through several cycles that are regulated by a special family of proteins, and it controls the progression of a cell through the cell cycle by activating cyclin-dependent kinase enzymes, or groups of enzymes that are required for synthesis of the cell cycle. And so when we talk about the cancer cells, they are essentially cells that have gone wrong. So they no longer respond to many of the signals that control cellular growth and death. And cancer cells originate within tissues, and as they grow and divide, they diverge further and further from that normal state. And so over time, these cells become increasingly resistant to the controls that maintain normal tissue. So they divide more rapidly, and their progenitors become less dependent on signals from other cells. So they become more and more disconnected from the rest of the organism. And then they evade programmed cell death, despite the fact that their multiple abnormalities would normally make them prime targets for apoptosis. And in the late stages of cancer, cells break from normal tissue completely. They go outside of the boundary of that tissue and eventually metastasize or spread to new sites of the body. And so to help us think about this process of cell deregulation, we can think of the different hallmark features of cancer. So as we said, cancer is unchecked health growth. Mutations in genes can cause cancer by accelerating cell division rates or inhibiting normal controls on the system, such as cell cycle arrest or programmed cell death. And as a massive cancerous cell growth, it can develop into a tumor. And so when we think about these hallmarks, essentially, there are original 6 hallmarks of cancer. 1, sustaining proliferative signaling. 2, evading growth suppressors. 3, activation of invasion and metastasis. 4, enabling replicative immortality. 5, inducing angiogenesis energetics. And 6, resisting cell death. And so in the recent time, additional 4 have been added, such as deregulating cellular energetics, avoiding immune destruction, tumor promoting inflammation, and genome instability and mutation. So here we observe the process of a healthy cell that eventually becomes an invasive tumor. And so we see this as a complex condition that manifests in changes to cell regulation at many different levels, resulting in this abnormal growth that we call tumors. These tumors consist of cells whose growth and morphological characteristics are very different from normal cells. And so criteria for malignancy would include some of those features that we just talked about, like cell proliferation, loss of differentiation, infiltrative growth, and metastasis to other organs that we associate with different stages of cancer. And so here you can see how the cells develop over time. And one of the primary objectives of oncology is to understand the mechanisms behind such processes that cause cancer cells to emerge and to grow. And so when 30 years ago, scientists could not even offer really a coherent answer to this question about what is the cause of these changes. And they just knew that cancer arose from cells that began to all of a sudden proliferate uncontrollably within the body. And they knew that chemicals, radiation, and viruses could trigger this change. But exactly what happened, nobody really knew just 30 years ago. But over the last 3 decades, an emergence of data about these cells has revolutionized our understanding of cancer. And this is because we have different techniques now of molecular biology, as well as data science, that allow us to generate data from the DNA and the RNA and the proteins in these individual cells, tissues, and organs, and associate them with specific molecular factors that could help us understand how this process actually evolves. And so when we see these cancer cells, we know that they are very different, right? So for example, how can we describe this difference? One of the ways to describe them is using phenotypic data or phenotype. And so we can look at their morphology and how they look and kind of try to categorize them. For example, they have large, variably shaped nuclei. They have many dividing cells that are disorganized and they're not arranged in the same structure as a tissue. They have a lot of different shapes and sizes, and they essentially lose the normal features that we associate with cells that have a very defined structure and shape. And so as a result of observing these phenotypic changes, we can start asking the question, well, how is this phenotype as a result of a disrupted production of cells, which is regulated at different levels of DNA, RNA, and signaling? And so we can then start to understand some of the basic internal cellular factors that contribute to the development of cancers. But what we know today is that the cancer cells inside the tumor also leverage some of the cells in the tumor and around the tumor. And so we can think of this as a disease that affects the tumor cells as well as the tumor microenvironment. Because cancer cells are able to ignore signals that normally tell cells to stop dividing or that begin a process known as programmed death or apoptosis. And so the body uses this process to get rid of unneeded cells. But because they stop responding to these signals, they also start affecting the cells around them. And so one of the things that we have to study is the cellular mechanisms, the interaction between cells within a tumor that actually contain many different cells, but then also to think about how do they interact with the rest of the tissue, the rest of the organ, and eventually what causes them to go beyond the tissue itself, causing what's called invasion, infiltration of other tissues, as well as metastasis. Now, one of the other hallmarks that we talked about was the evasion of the immune response. And so when we think of cancer cells that stop responding to the regulation within the cell, there are specific types of immune cells that are supposed to recognize those and attack them. And so cancer cells are known to evade this immune system, which is a network of organs, tissues, and specialized cells that protect the body from infections and other conditions. And so normally the immune system would remove damaged or abnormal cells from the body, but some cancers are able to hide from the immune system. And so there are two types of tumors, hot and cold tumors. What are hot tumors? Hot tumors are those that show signs of inflammation, meaning the tumor has already been infiltrated by T cells rushing to fight the cancerous cells. So the tumor cells have undergone many mutations that create neoantigens recognized by the T cells, so they can recognize them as abnormal cells. Now, typically with the presence of cancer-fighting cells or T cells, we know that the immune system has recognized them and is working to fight. And so for this reason, hot tumors typically respond well to immunotherapy treatment using checkpoint inhibitors because those are present. And so the main idea behind checkpoint inhibitors is to use antibodies to mobilize even more T cell response. During the attack on the tumor, T cells become exhausted and lose important functions needed to kill tumor cells. And so the exhaustion is brought on by constant exposure to tumor antigens and signaling through checkpoint receptors. The antibodies block signaling through these receptors to prevent this loss of function. So we'll look at a project where we can observe this loss of function in these macrophages. What are the cold tumors? Some non-immunogenic cold tumors have not yet been infiltrated by T cells. And so this is a sign that the immune system is not working on for some reason, right? So the lack of T cells makes it difficult to provoke an immune response with immunotherapy drugs. And so what we see at the cellular level, to the tissue level, to the organ level, is that these different characteristics of cells at the molecular and cellular level can help us diagnose specific types of processes and try to address them using a number of innovative treatment options. So finally, one of the main reasons why cancer progresses to a late stage and causes mortality is because of metastasis. And so when cancer cells spread locally by moving into near normal tissue, that's called invasion, but then they can spread to nearby lymph nodes, tissues, or other organs. And so as a result, we see this metastasis of cells in the whole body. And so when you observe under a microscope, metastatic cancer cells have features like the ones that the primary cancer where they came from, and not like the cells in the place where the cancer is found. So you can have, for example, a metastatic cell come from the breast and go into lungs. And so that is essentially a breast cell that is causing cancer in the lung. And so what we see here is a number of insights that could be very critical for our ability to treat cancers and recognize them, understanding the reasons or the drivers of this progression. Okay, so any questions so far about what we have covered? If you have any questions, we recommend putting those in the chat box. So again, if anyone has any questions about the process that we described here at the biological level, if you have any questions about the program itself, you can use the chat box to write those out. Another resource that is important is that we have a Slack channel for those that have completed their registration to the Precision Oncology program. So if you put any questions that you would like us to address in our next session, please add those to the Slack channel with a tag question, right? So if we have a number of questions for our next session, we'll be able to reflect on the material that we've covered today and continue on to speak about the additional material. So what are we going to speak about? Okay, and so here we already have a question. Describe more about checkpoint inhibitors in immunotherapy. So great question. Now we have to speak about what are the methods that we have to identify these specific changes, how signaling or the genetic regulation of different types of expressed proteins happens within the cell. So let me talk about that. And Sakshan, if you are already a part of the program, please add your question to the forum on Slack, and we will start with that question in our next session. Okay, so when we speak about the ways, the methods to understand what is going on, one of the things that we have, one of the technologies that allows us to study these processes at the subcellular level, in other words, inside the cell, are techniques like next generation sequencing. So next generation sequencing is essentially the process of taking the genetic material from within the cell of multiple cells within a tissue, extracting it and converting it to digital data. So essentially we can survey what exactly is happening at a given snapshot in each cell. And this could be done using a variety of what's called omics data. So omic data includes phenomics, genomics, epigenomics, and transcriptomics. And omics technologies like next generation sequencing can be used to explore the roles, relationships, and actions of the various types of molecules that make up the cells of an organism. So for example, here you can see phenomics, right? So it's a collection of automated ways to categorize cells. For example, we talked about cell morphology, right? So this could represent some kind of a phenotype that we associate with a given cancer cell. Genomics, or the data that represents the sequence of the DNA. Genomic data represents either the individual nucleotides or the sequence rearrangements or the variation in segments like chromosome rearrangements. So when we talk about genomic data, we'll look at mutations, copy number variation, and rearrangement. Then we can talk about epigenomics, or the way that this data in the DNA could be silenced or made more available for reading using a variety of epigenetic regulation mechanisms such as small microRNAs, long non-coding RNAs, and other types of methylation and changes to the chromatin, right? So we'll talk about some of the role of the epigenomic data that can reveal those regulation patterns and mechanisms. And finally, we have the ability to measure expression levels. So we can ask the question, which proteins essentially are going to be produced by looking at their proxy, or the mRNA. So we can look at the whole spectrum of omics technologies using the sequencing technology of next generation sequencing and start asking questions. What is happening to the cell? What does that mean at the genomic level? How is it regulated at the epigenomic level? And what is the result of that regulation on what is being produced? And so as a result, what we want to do in this program is take a look at examples of basic and translational oncology research projects that will take a look at single cell patient-derived cell line projects, that will look at systems level patient-derived xenograft models, and finally look at real patient data where we can get data from clinical trials and a variety of translational projects that will help us understand the relationship between comorbidities, risk factors, individual people or patients, and how we can use all of that data together for a variety of translational applications. Okay, and I'll pause here just to answer the question. So what are cell lines? Cell lines are essentially cells taken from tumors of individual patients that are then immortalized and could be grown in a lab. Instead of in a body, they could be grown in a petri dish. And so you can see individual cells that represent essentially what happens in an organism to a certain degree of what we can tell about a cell. So as we gain access to these different data sets, there's also a variety of different challenges that are arising from this availability of data. So as you can see here on the graph, starting from the 2010, approximately, we have this dramatic increase in the availability of public data, but it comes with also additional challenges. One of the challenges is that this data is very variable and very specific to individual projects, essentially, that generate it. It's also huge. The data sets are fairly large. If you take one next generation sequencing data set that might have between several to maybe hundreds of samples, you're talking about gigabytes to terabytes of data. And so what's important about being able to handle such data, to process it, is that in this program, we want to introduce you to some resources that will allow you to perform computation of huge data sets using cloud-based resources and leverage them to essentially develop structured data out of these publicly available data sets. So how are we going to do that? In each case, when we talk about genomics, epigenomics, transcriptomics, proteomics, we're talking about data sets that are contained in packages of short reads or elements that have to be pieced together to represent a structured data set that we could actually analyze. And so one of the things that we will do in this program is we will take this raw data. So what you can see here are individual reads mapped to a reference genome. And so we'll look at this raw data to try to understand how we can detect in this raw data specific signal. And that signal could be variants, so specific positions at the DNA level that have a mutation or a change. We will look at the processing, quantification, and performing statistical analysis of mRNA expression data, including processing that data from raw reads to a table of expression and asking the question, how can we compare different conditions to see changes within specific genes? And we'll also think about how do we leverage big databases where we can use annotation on gene ontology terms and pathways so that we can understand how this information, the signal that we find in the data, could be turned into biologically interpretable information. Okay. Okay, and so we'll talk about the schedule here in just a second. Thank you, Sylvia, for the question. But let me first show you how we can use this resource for big data analysis that will allow us to essentially study such data. So please go with me to a link server.t-bio.info. And Beepsa, if you could please post the link as well as the demo details. So this is a platform, a cloud-based platform for big data analysis where you can analyze large data sets from public repositories like the NCBI or TCGA, the Cancer Genome Atlas, by uploading data directly from these public repositories or using data sets that we'll be providing you with. These are curated data sets that you can use to perform analysis yourself. And so the first thing that we want to do is look at one demo pipeline. So this is a pipeline that will illustrate how the system works. And you will see how logic of analysis will be clear to you after just this one exercise. Okay, so please put number 1 in the chat box if you have gotten to this platform and you see what I see on the screen. Okay, so please put number 1. I'll just give it a moment for everybody to get here. Okay, thank you, Rajneesh, Norman, Musumi. Okay, let's give it just another minute for a few more people to get to this link. Now, once you get here, you can log in either using account details that have been provided to you by email for those that have registered to the program. Please make sure to add marketing@PineBio to your contact list, because you will be receiving emails about important account information after this session. Or you can use the demo account to log in. All right, great. So we have a few people that are already here. So right now, if you don't have an account, you can use the demo. So right here under transcriptomics, you want to click on this arrow. And let's go to this first one, Demo RNA-seq Cell Line Project. So once you click on that link, you will see this pipeline. This pipeline combines a number of steps organized into logical groups by what you see in this frame, like error correction, exon detection, mapping on genome, etc. And now what we'll do is build a pipeline. So we'll essentially connect a series of scripts together to convert data from raw format, which is individual reads, into a structured format, which is a table of expression. So first of all, I will click on Start, and you can see how these logical steps are organized into a sequence. First, we do pre-processing, where technical variation is removed from individual read sequences. Then we perform mapping, where you align those reads to the reference genome. And then we quantify the levels of expression. So we ask the question, how much of a given mRNA is present in the sample? Let's click on OK. As you can see, I have a number of options that are now highlighted: Trimomatic, bowtie 2G, top hat 2, HiSat 2, and bowtie 2T. So based on the logic that we just discussed, pre-processing, mapping, and quantification, what do you think should be the next step for me to take? Which button should I click on next? Remember, it was pre-processing, mapping, and quantification. Which one should I choose? Pre-processing. Okay, so which step specifically, what does that button say on it? Which button should I click on? So as you can see, pre-processing, we've correctly identified as a whole. Okay, great, Saksham. Trimomatic, right? It's the only one here. So let's click on Trimomatic. Okay, and let's learn about what does Trimomatic actually do. How is it a pre-processing step? So Trimomatic, you can scroll down here and it says Trimomatic algorithm cleans technical sequences from a database which stores sequences known to be used as adapters in next generation sequencing experiments from raw sequencing data. So what is raw sequencing data? Individual reads. And what sequences are present? There are specific sequences that will align to a database. And we know that there are going to be technical sequences, adapters, and sequences that essentially all of these will be trimmed, right? So we go through all of our individual reads and trim sequences that are not representative of the true biological signal in our data. So this is Trimomatic. So after we click on Trimomatic, we have another one pop up, which is called PCR Clean, which addresses the step of this bridge amplification. We'll talk a lot more about this in our next session. And after we've done some pre-processing, we now have mapping on transcripts. So essentially, we're going to map those reads onto a reference transcriptome. And then we will produce a level of expression or quantification. So we will quantify for each gene that we have in the human genome. For example, we will give it an expression level. And so using this RSEM method, that will produce a table of expression. We can now click on End. And if you just wait a little bit, you will get here some of the outputs from this pipeline. So please go ahead and explore this pipeline, the outputs that we see here, to understand how processing will help us produce structured data on thousands or hundreds or tens of samples and thousands of genes. So this is something that we'll use in the context of specific projects. So you probably saw that the project data set that we have there is called cell lines data. And so together with the cell lines data, we have a variety of different projects, as you can see right here. And we hope that in the process of this program, you will also develop a project of your own. So learning from these examples that are taken from top peer-reviewed publications in application to cancer, we'll see how you can develop a project of your own or potentially add on to what's already in the data set that is described here. And so here we can see some of the examples at the level of predicting response or outcome for different cancers, discovering novel ideas and hypotheses about the risk factors, and even potentially developing novel intervention methods. And so just to give you a sense of how this will be done, here's an example of one of those projects called Modeling Precision Treatment of Breast Cancer. This is a data set from a publication that was published in 2013. And it's interesting because here the authors assembled a collection of samples with multi-omics data that could help predict drug response. In another project, as you already heard Dr. Harpreet Kaur describe her experience with the Cancer Genome Atlas, we will go to the Cancer Genome Atlas, which has samples from thousands of patients across a variety of tumor types, and we'll start asking the question about specific types of clinical information that we have and phenotypic information that we have about those patients. And we'll go into the publication to understand what novel information has been extracted from this collection of patient data associated with molecular data that can help answer questions about specific aspects of hepatocellular carcinoma, like drivers of hepatocellular carcinoma, different stages and biomarkers of subtypes, as well as some of the response to different treatments. So what we'll do in this program is we will talk about cancer biology, including the central dogma of molecular biology, the biological hallmarks of cancer, the approaches to cancer diagnostic and treatment selection, to the understanding of the biological factors of the tumor itself, as well as its microenvironment and heterogeneity, and the regulation and targets in specific biological pathways that could provide us with a good understanding of biological factors associated with this disease. Now, we'll also talk about bioinformatics methods like processing next-generation sequencing data, variant calling, the application of RNA-seq analysis, statistical analysis like hypothesis testing and regression, machine learning, including clustering, principal component analysis and classification, and annotation of gene set enrichment, gene ontology terms, and KEGG pathway. And then finally, we'll introduce you in detail to curated data sets from these exemplar projects. And these include projects from cell lines, the patient-derived xenograft models of tumor and stroma interaction, where a tumor is engrafted into an animal mouse model, the Cancer Genome Atlas, or TCGA, that will allow us to think about clinical risk factors and outcomes, and also think about different types of therapeutics for different subtypes and stages of cancer. But we'll also talk about how to find additional data on NCBI and TCGA and how to structure your bioinformatics project. So now to answer Sylvia's question and to talk about the program schedule and how to gain access to all of these resources, I will pass it back on to Bibsa Biswas to talk about the program schedule.
Thank you, Elia, for sharing this wonderful overview of what we are going to be doing in the upcoming program. And now let me introduce you to the OmicsLogic resources that we will utilize during this entire training period. The program will leverage our top-rated online tutorials using the learn.omicslogic.com portal with smart tracking activity and curated datasets designed for great learning experience. And throughout the course, you will be given directions to access the TBio platform for bioinformatics processing and analysis of RNA-seq data. The platform will include demo pipelines like we saw today, as well as data management and analysis cloud infrastructure to run bioinformatics pipelines. Different stages of analysis will be performed in different sections of this multi-omics platform. And in this program, we will be using a research bioinformatics analysis to help you develop a logical understanding of key methods involved in preparation of such data. And behind many of the methods are also scripts written in popular coding languages such as R and Python. These are also explained in tutorials you will have access to in form of the Code OmicsLogic platform, from visualization of genomic data to exploratory analysis and hypothesis testing. And if you have not checked out the Precision Oncology link, here is a snapshot of all the sessions that we will cover. So to answer Sylvia's question, We are going to be having sessions on Monday, Wednesday, and Friday at the same time, which is 10 a.m. CST. And if you are looking forward to join the program, this is the link. So let me take you back to the website again and show you how you can join this program. So this is the link to join the program. I will be sharing the link in the chat box. If you have not yet registered for the program, the registration is still open and you can go ahead and see what the program is about. We have also added the different dates and the session topics here, so you can actually see what is the next session that we are going to be having. And to register for the program, once you click on Get Started, I'll go there again. You will find there are 3 different licenses. So as we are beginning, I'll just again explain what these 3 licenses are. The beginner license gives you access to all of these resources that we discussed today and the sessions, as well as the Slack channel and the project resources. While the intermediate license gives you an access to a mentor who you can meet one-on-one every week to discuss any questions that you may have or to discuss your project. While the advanced license gives you an additional 30 days, sorry, not 15 days, additional 30 days to work on your project with our team of research fellows, as well as collaborate with any participant. So as you know that the program is ending on 9th of April, and we will give everybody an extension till April 16th. Post that, from April 16th to May 16th, anybody with an advanced license can actually go ahead and work on their project. So they will continue with their access to the resources as well as the program. So I hope that this would be clear for everybody. So let me go back to the presentation. I hope that you have been able to review all the different dates for the session as well. And now I would like to invite Clinton to talk a little bit more about what is the outcome that every participant will have after they complete this training. So Clinton, over to you.
Thank you, Viksha. So, as seen in some of the earlier presentations, we will learn important concepts within cancer biology, such as the central dogma, the hallmarks, how cancer is diagnosed and treated. We will talk about several bioinformatics methods, such as processing, variant calling, RNA-seq, and more. Finally, we will look at sample projects to learn how to make your own projects. By learning these concepts, you will be well structured to start your own projects for future analysis. So the learning outcomes is as follows. You will learn to read and understand bioinformatics literature, perform basic NGS analysis, form hypotheses related to bioinformatics questions, prepare data for machine learning, cross-check actual signals or actual data with other gene databases, know what type of data is required to answer those scientific questions, create your own bioinformatic analyses / pipelines, finally present your results in an organized way. All these skills are essential in bioinformatics, and some can be translated to other fields. After achieving the learning outcomes, you would have learned some of the major best practices in the field of bioinformatics. You will be exposed to R and Python, 2 powerful programming languages. Bioinformaticians will need to understand how to use t-tests, regression, and other tests. Analyzing and managing data, you will also learn how to analyze and manage data in order to communicate important information to other individuals. Pipeline development and utilization is another common best practice within the field of bioinformatics. Ultimately, completing the Precision Oncology program will help you enter the field of bioinformatics, since you will learn several best practices. So now I would like to pass it back to Deepsha to talk more about the registration and form.
Thank you so much, Clinton. And the chat box is now open for any questions. And while we are doing this, I would want to know more about everybody who is present here in today's meeting. So now you'll find a poll on your screen. I hope everybody can see the poll. And there are a few questions that we have for you. The first question is, what is your current country of residence? And if you are from any of the other countries which are not listed around here, you can also post them in the chat box. The second question is, what is your current level of education? Again, if these levels of education are not mentioned, you can also mention the others right in the chat box. What is your area of interest or specialization? Again, you can also write about any other area of specialization which is not pertaining to oncology. And the fourth question that we have is, what are your objectives in learning about precision oncology? So there are a few topics to choose from. If you are a beginner and you have an interest in cancer research, so you would want to learn more about precision oncology. You already work on cancer research and want to implement bioinformatics to your research. You find the topic interesting, or you're not interested in oncology at all. So you have all of these options to please go ahead, and I'll keep this on for 1 more minute, and then I'll go back to the presentation. And meanwhile, as you are going forward with the poll, please make sure that you also post, you also post the questions in the session. Yes, and I think David, I'm sure I'm getting your name correct. So we did have the introduction to oncology at the session. Did you join in earlier? I did not see your message. And as you're joining in, if you're participating in the session, what would be great is for us to know more about who you are. So if you can always have your full name on, that would be really, really helpful. Okay, so I will be ending the poll right now that you see on the screen. And next, again, what we would want everybody to start with is sign up to learn.OmicsLogic to get started. And you know, we have already spoken about the Slack platform that every participant will have access to who have joined this training. So you can post any of the questions that you have regarding the session right there. As well as we have our LinkedIn page and our Facebook group that you can also post in. And once you complete the training, we will also give you a certificate like this. And these are some of the details regarding the training program. And we have already gone through this one. So please feel free to check out the link on how to join this training. So in our previous program, we have seen participants who have completed the training successfully, like Olga Udatseva here, who is a postdoctoral research fellow at Roswell Park Cancer Center, and used her experience to learn about data analysis techniques, which she's now applying in her own preclinical research. And like Olga, there have been several other participants who have taken part in different OmicsLogic programs that we offer and have been very successful at them. And once you complete the program, you will receive a certificate of participation that you can use on your resume or link to your profile that shows all of your achievements. And this is why our previous program participants like Mikhail here ranked the OmicsLogic programs as a top based on program interactivity, personal feedback, support, and the quality of projects you will be able to achieve leveraging the resources we make available. And Mikhail has shared this very interesting review. So I will again go back and share this review with everyone here, so that you all can also take a look at what he has to say about the OmicsLogic programs right now. And while we are here, please make sure that if you are joining the program again, this is the link to join. And we just want to mention that we also offer scholarships for students who require them. So if you are someone who is interested in applying for a scholarship, so please go to this link right here and you can apply for the scholarship. So once you apply and fill up the form right here, you would receive an email from our end where we would ask you to set up a meeting with one of your references. So based upon that meeting, it is decided whether you are going to receive the scholarship or not. But please feel free to go ahead and fill out this form right here if you are interested in applying. And I do hope that you have, if you have any questions, you'll be posting them in the chat box. So in the upcoming session, we will discuss the types of information that can be extracted from next generation sequencing data. And you will learn about how to process, analyze, and interpret genomic data through specific pipelines. Example techniques will include variant calling, mapping, and finding the significance of specific mutations. These are the common techniques used in hospitals to analyze sequencing data from cancer patients in hopes to diagnose and potentially design an effective treatment plan. With this skill set and a few project worth of experience, you could also be potentially on the path to help a hospital near you or become a bioinformatician who works on cancer research. So now the session is again open for any questions that you may have. Please feel free to ask us any questions. Okay, and you can also see the poll results right now on your screen. So I see that we have about 8% of people who are from the US, 23% from Mexico, and 8% again from Peru. We also have a large number of participants today who are not from US. So we also have here in the chat, Nigeria, Iraq, and Bangladesh. What is your current level of education? So most of the participants here hold a bachelor degree or are in the undergraduate program, while 23% are in master's degree, 15% in PhD, while 15% are also faculty, while there are others who also are compromising of 15%. And area of interest or specialization. So 54% present here are, of course, well into precision oncology or oncology as a whole, while 23% are interested in bioinformatics. The rest are undecided or interested in something else. You can also write about those interests right down in the chat box. What are your objectives in learning about precision oncology? So you're a beginner and your interest is in cancer research. 69% have said that they are a beginner. I work on cancer and want to implement bioinformatics. So that is comprising of 31%, while we do not have anyone else for the rest of the fields listed there. So thank you all for sharing again. Again, if you have any question, we will let you know. So Tolulope, are you already registered for the program? So this was the only free session that we have as part of the Bioinformatics for Precision Oncology program. For the upcoming 1-month program, you would have to register for the full program to gain access to those sessions. All participants will receive an email, and the sessions, please note this, the sessions will start at 10 a.m. CST. So if you have missed out or joined later, please review the video that we will be emailing you and sending it to you. And we will also share those videos in the Slack channel so that you are able to review them later as well. And if you have any other questions, please let us know.